Good evening, everybody. So my name is Kati Steppe. I'm from the Faculty of Bioscience Engineering in Ghent in Belgium. But before I start, I would like to thank Uri for inviting me here. It's a pleasure. It's also my first time in Israel. I will talk a bit on tree hydraulics, but with a flavor of carbon, let's say. Okay. Oh, I have to speak here. Okay. So if you look worldwide, then we know that trees are under pressure. They suffer from a lot of things. They are threatened by and they must survive a lot of stressors, and one of them is drought stress. And depending on the level of drought stress, the trees will be healthy, can be unhealthy, or can be in a transition phase. And the challenge, I think, of some of our current generation is to predict how trees will respond to climate change. And therefore, we need to determine, can we predict indeed this transition from healthy to unhealthy? Can we detect signals? Can we detect signs that show that a certain threshold will be crossed? And so therefore we need early warning signals, because there is a saying that says only those who can see the invisible can do the impossible. And driven by this idea, the ambition of our lab is basically to listen to the plant's heartbeat and to look for these early warning signs. And to do so, we equip plants with a lot of plant sensors. So a lot of our experiments in the lab looks like intensive care units, but then for plants. And by putting these sensors on the plants, we turn them into a biological indicator. So the plant can tell us the story through the signals. And as a bioengineer, we translate then this signal and try to translate the story that they are telling us. And this technology we use in an, let's say, worldwide network. We are aiming for that. It's called Tree Watch, and so there trees are equipped with sensors and with the idea to indeed give us this early warning signals. And by monitoring trees, we can look at their dynamics. We can look at their dynamics in water relations. We can look at their dynamics in carbon relations. And you see here the series of sensors that we can apply continuously on the plant itself. And then we can understand their behavior. We can understand their functioning, both in the water relations. So we can look at their subflow. We can detect hydraulic resistance, change therein. We can quantify hydraulic capacitance with these sensors. And if we look at the carbon side, we can look at growth, but also to respiration and what happens with the CO2 that evolves from respiration, and that will we deal with after the first part. Okay, so if we have a tree and we equip it with a subflow sensor, then we can have continuous data of subflow, which gives us temporal dynamics, responses on the environment. And if we then integrate the area under this graph, we can quantify how much water the plant is using. So that means that we know the consumption of water of this plant, and that gives then also the idea for plant-based irrigation. How much should we give back to the plant? But these water dynamics, if we, if we put a second sensor together with the subflow sensor on the tree, then we get, besides the water dynamics by the subflow sensor, we also get stem diameter variations. And that means that also those give like heartbeat of the plant, let's say. So if the transpiration is larger than the uptake, then the tree will shrink. if the uptake is, I think something wrong. Yeah, if the uptake is larger than the loss through transpiration, then the diameter will swell. But you see that the swelling, and so the replenishment of the water of the tissues that were emptied before, it is basically larger than what the starting point was. So that means that another process happens during the night, and that is growth. And we can describe that mathematically by Lockhart's equation. So we can have a volumetric irreversible change by water. So if water increases the cell volume, then there will be an irreversible change in that volume, and therefore we see that during the night, when there is no sap flow, that we have this growth happening. And these patterns, these dynamics, these signals can be used to inform, or can be used to inform us as how the plant is feeling. So I will illustrate this because if you combine this set of sensors with mechanistic models, so you can describe all these processes that are ongoing, so these dynamic processes, you can describe that mathematically in a mechanistic way. Ah, that's maybe easier, yeah. Okay. So if we describe that in a mechanistic way, and we combine that, then basically we can develop advanced control algorithms. So based on the signals that the plant is giving us, we can take then an action. And I will illustrate that in this case, that is indeed much easier, by a diameter variation model that we have developed and that can be used for example, plant-based irrigation. So here you see, again, data from well-watered plants. 4 days of data are collected. And if we then project there the model results on it, so we use these data to calibrate the model, then we see that we can predict quite well the measured data. What we then do is we take the environmental input and we can predict the next day with the model, with an error band. So this is what the plant should do with the environment that it gets. If we then compare that with the measured data on that day, and it stays, like in this case, within the error band, and we know that the plant has performed like we said that it would have been, right? And then the cycle repeats itself, so we have a moving window. So that means that we calibrate the model again, we can predict again the next day, and we can compare again the measurements with it. So that means that you have an online tool to see whether or not your plant is behaving like in normal and well-watered conditions. So now, if you have then this tool, then you can also predict when the plant is not behaving well. And for example, here you see 2 days where the measurements go out of the error band of the model. So that means that the plant has stress because it was not behaving as in well-watered conditions. And so that gives then a signal and can be used by farmers, for example, to irrigate then. And the 2 triggers that you see here was due to high air temperature and low air humidity, so basically a high VPD. We can do the same thing for salt drought. So the first example was atmospheric drought. If we go then for salt drought, then you see that indeed here the plant was not watered. You see the dynamics, and you can also use the measured data already to see that the plant is indeed not growing very well anymore, that also the shrinkages, so the black line I'm looking now at, that the shrinkages are increasing. But if you again use the model, you can basically, as a non-expert, also see when the measurements deviate from the model. And then you see that if we look again to this model that goes outside of the error band of the measurements that go outside of the error band of the model, then you see that we can already detect the stress quite early. It is up to the moment that we water the plant again, then we even have a beneficial effect, the blue part here. So the measurements do better than what the model predicts. And in that sense, we have a cool tool that can be used to predict stress. So these early warning signs are given. Also, if you compare that with the traditional, like maximum daily shrinkage measurements that you can derive from this diameter variation pattern, so the daily shrinkage you can calculate, this tool gives even 2 days before that already an indication that something is going wrong with the plant. So based on these measurements and the models, we can have early warning systems that can be used. Okay, so to come then to the topic of and the title of my talk, what has CO2 to do with that? Well, in the climate change world, we of course need to understand the effect of CO2 on all these processes. So what you have seen till now is under normal atmospheric CO2 concentrations, but what if CO2 increases? And I will then end not only with what if the CO2 in the atmosphere increases, but what is happening with the high internal CO2 concentration in the stem itself, and that whether that is true or not, I will directly indicate. So first of all, the effect of elevated atmospheric CO2. For that, we had 2 treatment chambers, climate treatment chambers, where we had 400 ppm and 700 ppm on populous tremula trees, so poplar trees. And so these trees were measured with a lot of sensors, but for the talk I limit myself now to stomatal conductance measurements. Here you see again another type of dendrometer for stem diameter variations and water potential. What was the setup that we studied is we looked at drought and well-watered trees, and then we had an additional batch that were well-watered trees and that we took to the lab for then bench dehydration. So that means that the trees were well-watered, they were grown under 400 and 700 ppm, and then on the bench they were dehydrated, and as they dry out, basically what happens, the embolism formation sets in, so that means that we get air bubbles inside the xylem column that will have an explosive evaporation. So that means that we will have a pressure wave in the water potential going from a certain negative water potential up to positive values. And this pressure waves basically creates a sound. And sound is something that we can measure, that we can detect with acoustic sensors. So this is basically such a broadband acoustic emission sensor. And every time that such an embolism is formed, you see here all these dots, so that is through dehydration, and each dot represents a signal. And then you can either work with just the clicks that you hear, or you can also analyze the information coming from the sound to have additional information on, is it shrinkage, is it coming from real embolism formation. So there is a lot of information in this acoustic technique. In addition to that, as you can see, we also measure then, again, the dendrometer to have an idea on the drying out of the plant. So here you see a bit as a visualization. So you can put the tree indeed in a micro-CT scan. So also scan that if we break a bone, it's the same technique, but then in a higher resolution. And then normally all the vessels will normally transport the water. At the moment we have drought stress, then these water-filled vessels will translate into like here, air-filled vessels, and that gives then or results in the clicks that we measured with the acoustic emissions. We did this experiment in 2 parts of the season. So we had a drought stress treatment, and once we did it in early season, and the second time in the late season, but in year-wise it was the other way around. So we started with the late season experiment in 2018 in summer and autumn, and then we continued with the early season experiment in spring and summer of 2019. Okay, what was the result with these acoustic emissions? Here you see the vulnerability curves that come out of these sensors, and then what you nicely can see is that the elevated CO2 curve, which is this one, that so elevated CO2 makes the trees more vulnerable to drought stress, because here on the x-axis you see the water potential. So the more negative the water potential becomes, the that gives them the signal when there will be 50% of loss of hydraulic conductivity, here expressed as cumulative acoustic emission signals. And you see that indeed in these elevated CO2 that happens faster than in the control trees. Why is that? Because under elevated CO2, we have fast growth, and that changes the extensibility of the cells and makes them weaker for this embolism formation. If we now do the same experiment in late summer, then also under elevated CO2 and atmospheric CO2, you do not see that effect. And that is because later in the season, then the CO2, the extra CO2, is basically formed to have cell wall thickening. And therefore, the effect of the weakening of elevated CO2 is eliminated here. Okay, if we, in that same experiment, look then at stomatal conductance, then in this experiment, we did not find any significant result on the well-water trees of the elevated CO2, not in the early drought and the late or the late drought treatment. But when we looked at drought stress, so if we subjected the trees to drought stress, then you clearly see an effect. And if we combine that then in cumulative curves, then we saw that the elevated CO2 treatment, which is now this full line here, that the stomates of the elevated CO2 trees, that they close faster than the atmospheric CO2 plants. And that is kind of logic, because the ones that grow under elevated CO2 are more vulnerable to drought stress. So that is, or might be, one of the reasons why this pattern happens. If we do then, and we compare it with the same setup for a late drought stress experiment, then basically statistically we only saw a shift in the inflection point, but and it could be that the sensitivity of the trees to this elevated CO2 was either influenced by phenology, by being towards the end of their growing season, or something in the pathway of abscisic acid could also be a reason. Okay, and then if we go then to the diameter variation sensors, so in case when you have drought stress, you saw in the earlier experiment that the tree will not grow anymore, and then you can look at this daily pattern and take the minimum and compare it to a value where the last growth was detected, and that is what we call tree water deficit. And so if you quantify that again for the treatments, then you see in the well water treatment we saw a very nice growth in the 2 experiments. Here you see a bit flattening off of the curve due to the phenology. But as soon as there is drought stress, all trees basically they stop growing after a certain moment in time, and then you see that the this tree water deficit increases in function of time. So that gives also the opportunity to create these kind of curves. And there is basically on this parameter not an effect of elevated CO2. But what does this parameter tell us? That is basically the water that can be used from internal reserves, from living cells, to contribute to the transpiration stream. So that is the last, let's say, detector. And if we bring then everything together, all these curves that we have made for early and late season with these different types of sensors, then you basically see first that the stomates respond. Then it is continued by the dendrometers that gives the water that is coming from the living tissue. And then basically we have the acoustic emissions. And okay, they have an influence on the change in hydraulic resistance, but at the same time, they also are they supply water to the transpiration stream, so they are also a capacitor. So that is the third way of supplying water to the transpiration stream, and it is always the sequence that we see. And that means that if you have these kind of curves, that closely corresponds to what we need. So we need indeed these thresholds, these warnings, when does a plant go from healthy to non-healthy. So this is a first step towards that. And then you see that here in, if you compare the stomatal conductance and, the stomatal conductance and the acoustic emissions, then you see basically that the stomates are almost completely closed, and it is only at that moment that the acoustic emissions start to evolve and increase rapidly. So the water coming basically from these internal reserves from the embolized vessels, they set in later. Okay, so that for the outside, the atmosphere, the atmospheric CO2 concentration. But we can also have a look at internal CO2 concentration. And to give you an idea on that, so here you see, for example, some sections from a platanus species. And if you look at current growth branches, then the whole cross-section is full of chlorophyll. And the older the sections become, so 1 year old growth, then you see that there is still visibility of this greenness, but it's already a little paler. And the older and larger the stem segments become, then it's basically more or less more the edge of in the bark that the chlorophyll is located. So that means that we have green tissue there. And we can then wonder, what is that green tissue doing there? Well, and therefore we need to reflect a little bit on respiration, because respiration is the process that generates CO2, right? So, and what happens with the CO2, either in the night, most of it effluxes, so that is what you see here in this arrow. So during the night there will be efflux, but during the day, if you have green tissue, as we have shown here, and we have CO2 and we have water that is flowing in the stem, we can have local production of sugar. So this is basically what we call this woody tissue photosynthesis. So it's a local production of sugars in the stem itself. So these sugars are not coming from the leaves, they are produced locally. And that might be interesting. So we decided to play a little bit more with that feature. And here you see from small Populus tremula trees again, you see in function of the time, well, what we did is we packed basically the trees, and maybe I should go back, because here you see indeed the packing. We did it for small trees, but also for 4 meter large trees, and a team building event in the lab for a couple of days. So, and then what you see is if we do that, this is for, let's say, 1.5 meter large trees, and what you see then is if you do not allow the tree stem to have this stem photosynthesis, that they grow 50% less. So that means that there is a lot of contribution coming from this woody tissue photosynthesis. Surprisingly, if we do the same experiment at elevated CO2, then this effect is gone. Then these extra growth is not necessary, probably because the leaves are contributing sufficiently. If we then look to the vulnerability of the tissue, then you see if you pack the plant, that the plants become more vulnerable to drought stress. So that means that also there, that is sugars that are locally produced have an influence on the hydraulic integrity. And so we decided that we wanted to quantify this stem woody, woody tissue photosynthesis. And how did we do that? We measured basically the trees with cuvettes that were covered and uncovered. And here you see the measurements of an uncovered one, and we measure efflux. That means that we have lower efflux because the photosynthesis is possible to ongoing, so we have capturing of photosynthesis, while if you pack the stem, you do not have the woody tissue photosynthesis, and you have this signal, and if you subtract both, you basically have a quantification of woody tissue photosynthesis in the same units as we have for leaf photosynthesis. If you do the same thing for drought, you see similar patterns and similar conclusion. And now this is a nice summarizing graph, because here you see during drought stress in function of water potential, the leaf photosynthesis and the woody tissue photosynthesis. And as you all know, leaf photosynthesis drops down from a certain point when the water tension becomes too negative. At this point, the leaves are shed, basically. So that means here, and what you see here is the woody tissue photosynthesis. So if the plants are well watered, the leaf photosynthesis contributes like 10 times more. But if they dry out, at a certain moment, they are equal. And if the plant does not have leaves anymore, in all situations where there are no leaves on the tree, then it is the woody tissue photosynthesis that prevails. And that can explain why we sometimes see resprouting. That's often said, yeah, and we thought trees were dead, but we see resprouting because they have this woody tissue photosynthesis. So to summarize, this CO2, external but also internal, impacts the hydraulics that we have seen. And to have the whole story, basically we need to monitor and study and understand these processes to come to our prediction models for the future. Thank you.
They just get more saturated faster with elevated CO2, internal emission.
That is certainly one of the things that will happen, that internal CO2 will impact and the functioning. But the change what we saw was due to the fact that we only saw a little bit change in this inflection point. It was not such a clear message, let's say. So it seemed that indeed, and because this internal CO2, also for the early season drought, you would have the same reasoning that it has an impact on there. But apparently, if you do it in the late season, this trigger, this impact changes. It's still for us also a bit an unknown, although you might take into account his internal CO2 concentration. Okay. No, branches, large branches. Yeah, yes, yeah. Yeah, so and basically you can go up to small stems too, but these were on from the 4 meters large poplar trees, large branches from, let's say, a centimeter thickness. Yeah, 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 yeah. Yeah, well, we saw some leaf shedding, but and then indeed at a certain moment when we could not measure the water potential anymore, we stopped the experiment, and we then just tried some for biomass, but did not follow it up further. Yeah, to really investigate leaf shedding. Ah, okay, good. I'm here till the end, so if you have questions, I'm available.
Okay, hi everybody. Yeah, I will talk not about tree mortality, but about tree desiccation, conifers in particular. You might have heard last year, 2021, there was a huge heat wave event in the northwest of North America, so US and Canada. And we used this event to test some hypotheses. And if there's anything I want you to take home as a message is this. So we are used to study tree ecophysiology this way. We have a goal, we have a question, a hypothesis. We go to the greenhouse and we start with different treatments. As Kathy showed wonderfully, that's the golden way, and it's good. But science has more than one way. And that's my main point today. If we can be flexible, we can get to more interesting insight sometime. Not necessarily the most highly scientific, but still insightful. And we learned a lot, especially in unusual times, like the COVID that we all experienced, and they call for unusual approaches. So just to remind us that a COVID vaccine was developed in under 1 year. If you ask people in pharmaceutical industry, they talk to you about 10, 15, 20 years. But when you want and you have the means to do, you can get to big results in short time. So sometimes it pays off to do something maybe less scientific, but on a bigger scale. And we had an opportunity. So first, what we talk about. So this heat wave was really unprecedented, and you can see the effect here. Actually, I thought later, you see the 2-meter air temperature anomaly that gets to 15 degrees Celsius here. It was not even enough to describe what happens around this area here or in parts of British Columbia and Canada. So this was something like 20 plus degrees Celsius than what usually happens in this time of year. This was late June, early July, and it was warmer than I think you can expect in most of Israel in August, in any time. So what we talk about, something like 42 degrees Celsius, relative humidity, I had the meteorological data, it went down to 23%, which can give us a vapor pressure deficit of over 6 kilopascal. And this is a value often reserved to hyper-arid desert, like south of here, actually. So this was, it was called the heat dome. It was so shocking to people and to ecosystems. We had at the Weizmann some guests that came and told us about the deeper physiology that the forest experienced and flux towers and all that. But as I said, another approach is just to take opportunities, and that's what we did here. So the star symbol here is just over Seattle to show you that the place that we are talking about. And the story goes that I started getting emails from a very nice guy. This is the guy. He's a professor at UW, University of Washington in Seattle, not in anywhere close to plants, but his hobby is to grow conifers. So his name is John Albers, and he owns and manages the Albers Vista Gardens in Bremerton in Washington, US, just off the coast from Seattle. And that's a big botanical garden with many species of conifers. And I've never visited there. I was not far from there. I lived in British Columbia for a while, so I know the area, and it's beautiful and wet and so on. But then the heat dome hit, so probably it would look more like that. He is still smiling though. So he came up with email and asking, I'm seeing some weird responses on my conifers. What does it mean? What's going to be? And so on. So in the days that followed the heatwave event, he saw needle browning, of course, and that's what you saw already in the first slide. And it was shown on multiple conifer species, but to varying extents. And that's what caught me. So maybe we have an opportunity here, and that's what we did. So where others see despair, like most of the locals, we see opportunity. It's like a mega field experiment for us. And here we have a big common garden experiment with dozens of conifer species growing at the same place, the same time, affected by the very same event. So we're talking about almost 100 different species of conifers. You can see the different names here. A lot of abies, some cedrus, cupressus, others, junipers, of course, spruces, picea, pines a lot, these pinus, more pinus, Serutoga, of course, sequoia dendron, taxus, tsuga, and so on. And the first thing I thought, since we know the species, let's map them by origin. And he could tell me the origin of the species. And immediately you can see some trends, at least. So conifer species from the Mediterranean and from Central Asia showed little damage. So the color here is for the needle desiccation extent, just by 20% windows. So 20 to 40, 40 to 60, and so on. And even we had a few cases of more than 80% desiccation. By the way, spoiler, at the end of this, something like 2 months or 3 months after the event, almost all trees survived, except for 4 species, those that were really heavily affected. Not all of them, but 4 species were really dying. And these are the species that he had. I didn't mention that for some species, he had different ecotypes even. So different like sources for the seeds that he used. So those were less sensitive. Central Asia, Mediterranean, where species from Europe, you could see a bit more damage in European species, Eastern Asia, and even more so in North America. Many species here are more sensitive, and that was the first thing to do. But then, since we work on tree hydraulics as a community for many years now, we have already collected some data for the species. And although doesn't necessarily mean that these specific trees were the same as the trees that studied in France or in Uzbekistan or wherever, it does give us some hint. So the main idea here was just trying to correlate between traits that we have data for and the result that we had in the field, in the common garden experiment, which is the botanical garden. So just take this desiccation level and try to correlate it to different traits. And that's mostly what we had at hand. We didn't have any other measurements. We didn't send people to do measurements there. That was not the plan. But just with the data we have, what we can learn. And just to give some context, and this is taken by a recent review that we did with Nate McDowell and many good friends. So needle browning under heat and drought can relate to xylem cavitation. This is here. So runaway cavitation is something that you would expect under some more serious drying. And meristem death, for example, this is also something relevant that causes the browning, so some dead tissue. And let's see what that gives us. So the hypothesis was that the extent of needle browning should relate to xylem vulnerability to cavitation. And not for all, but for a good number of the species, we had the P50 values. That's the percent of 50% of xylem embolism, right? So that's the value, the P50 value in megapascal, and that's the extent of needle browning. And you could easily split this graph into 3 different regions. So species with P50 that was lower than -6 megapascal were totally unaffected by drought. And I give you some examples here, like this Lebanon cedar that you can find if you go 300 kilometers north from here. This Arizona cypress, for example, which is very resistant, and it's used for afforestation in Israel, by the way. And then you had species with a bit milder water potential for P50, between -6 and -5. And then they had mild extent of needle browning up to 25% of the canopy. And then you had all the rest. So the species with, if you have a P50 which is higher than -5 megapascal, browning was up to 95%, but could be anywhere between 0 and 95%, which this is already showing us that some other processes are taking part. And then we also had different ages of trees. So overall tree age was about 12 years, so quite young trees. And we observed that young trees were more vulnerable than older trees. For Picea, for example, we had a few ages, and for Abies concolor. And for example, among 11 cultivars of Picea abies, needle browning decreased with tree age quite a lot. So that's not all the processes that we could look at. And although we don't know mechanistically if this was leading to the observed browning, but we tried to look also on things like turgor loss, which is also an important process. And also, since we know that soil root decoupling is important in tree response to drought, we tried to look at a relevant root trait. So how about other relevant parameters? This is like the turgor loss point that showed actually no relationship, as you can see here. I wouldn't go too much to that. But rooting depth did show some relationship. And it seems that if you have the 4 species with roots that were reaching more than 2.5 meter had 0 damage. And generally species with roots reaching more than 1.2 meter had damage of 40% or less. So there was some correlation here, but again, we don't know how representative this is to the trees that were actually in the garden. Okay, so to summarize, we saw a sharp divergence among resistant and vulnerable conifer species according to the xylem vulnerability, all of which, again, simultaneously exposed to the same extreme event at the same site. And we think that this is evidence to the key role of P50, xylem vulnerability to embolism. P50 could not be used as a sole predictor of needle browning. As we saw, it was quite a messy graph. And most trees survived, as I told you before, supporting the notion that embolism kills conifers at rates which are higher than 70%. And among conifer families, quite importantly, from cypress family and like junipers, cupressus, calitris are the most drought-resistant plant species on Earth. But also, even among the local northwest conifer species that were grown in the garden, some cultivars proved hardier than others. So we already have some indication that for the next afforestation under climate change, these are good potential to use. Thank you, and questions. Um, Yeah, so there are two different things here. The susceptibilities that embolism can happen, and also how much embolism is enough to kill a tree. Yeah, and that's a different question. And we know that you need more than 50% to kill a conifer, for example, something like 70% or even more. But at a certain point, this can turn what we call runaway cavitation. It can turn like a process that magnifies itself and lead to more cavitation. But it's true. It's one part of the story. Yeah. And also it's an indicator. We don't say that everything has to do with embolism. Nadine. Yeah, heat resistance is a good idea, and it should, because this was a very hot event. But then we ask the question, and maybe back to you, what rate should we look at for heat tolerance? Something like, I don't know, I don't know if there is enough quantitative information on heat tolerance among these species. No, of course not. No, yeah. Voilà.
Professor also from the Weizmann Institute on the Yakir forests, malresponse to VPD.
All right. So, good afternoon or good evening or good night. I don't know, it's getting late. Thank you for inviting us for this session. I want to highlight the fact that, in fact, I'm giving the presentation of Yakir Preisler, who did his studies with us, and he's now doing a postdoc in Harvard, and he keeps delaying his return to Israel, so this is why I have to stand for him. So he prepared this presentation. I only made some minor edits. It's also, I'm being spoiled today, you know, I'm speaking after a great talk of a former student, and now I present a presentation from another student. You know, I'm really having a good time. So Yakir, also we're trying to confuse you here, because Yakir Preisler worked with Dan Yakir, and we both worked in the Yakir forest. So I'm sure at the end you will know all the Yakir, which one is coming from where. Yakir's work was to address the question is, what is the role of VPD? Or more precisely, can we distinguish between soil stress and atmospheric stress, moisture stress? So we all know that the climate is changing, and we expect both stresses to increase. There is going to be drying, and this is Mediterranean is a hotspot. Instead of reduced precipitation, temperature is going up, which is going to increase even disproportionately the VPD in the atmosphere. And of course, we know what is the dominant one controlling and how to predict the response of vegetation to these two stressors. The sort of the dogma in the literature is that VPD is a dominant factor, and it reduces conductance and therefore gas exchange, productivity, and survival of plants, trees, and forests. And the question if this is really substantiated. I want to try, we start actually in this presentation in a larger scale, and this is a recent study comparative study with collaborators in Greece, and we compared two Aleppo pine forests, one in Sani in Greece and Yatir in Israel. And what we see is a sort of adjustment that reflects perhaps an avoidance from both type of stresses. So here's the seasonal cycle in GPP in Sani in Greece, and this is the seasonal cycle in Yatir, and you see this, there is some decrease in the productivity, but the major observation is this large shift in the seasonality from midsummer productivity, peak in productivity, to late spring peak in productivity in Yatir. And of course, the conditions here are avoiding much of the moisture stress in the soil, because this is a rainy season or the end of the rainy season, and also the humidity in the air is higher. But still, there is a sharp decrease towards the summer, and both VPD is going up and soil moisture is going down. You can see that the same change in seasonality happen in, also in the net ecosystem exchange and in evapotranspiration, also a big decrease because of the much reduced precipitation in the area. But what is striking in these two sites is to look at the response of ET, evapotranspiration, to VPD. And what we find in Sani is that actually there is almost a linear response of ET to VPD. VPD doesn't go to the high number that we observed in Yatir, but in Yatir, when it's combined with, I mean, this is a system that is essentially almost never in terms of soil moisture stress. And in Yatir, when you have very high VPD, but it's combined with soil moisture stress, you see a very sharp decrease in ET to almost no evapotranspiration in the summer. So it's already some sort of a hint about the hierarchy between atmospheric and soil moisture. Now, if we zoom into Yatir, to the ecosystem scale, here is again the seasonality in net ecosystem exchange with the very early spring peak and a very sharp decrease towards the summer in both CO2 exchange and evapotranspiration. And we know that both the soil moisture is going down. This is soil depth versus the seasonality. And you can see here the depletion in the soil moisture at this period. But we also know that VPD is going up at this time. So this is again the seasonality, not exactly the same scale, but more or less you can see that both stress are combined already early in the season. So we have a problem of soil supply and problem with atmospheric demand, and what Yakir was trying to check out is which one is the dominant and the one that we have to be more concerned with. So he then started to do some physiological measurements in Yatir, looking at subflow, looking at gas exchange with, I think, 16 automatic chambers that worked continuously, measuring gas exchange and transpiration, and these are some of the results that he showed. So again, the first observation, a very dramatic indication of adjustment in the trees, sort of avoiding both stresses. So in the wet period, you see a good agreement between the subflow and the transpiration. The red is the subflow and the blue is the transpiration from the chambers. And you see a temporal agreement between the two, peaking around midday. But in the summer, in the dry season, there is a big shift in the transpiration to early in the morning, but not such a shift in the water uptake from the soil, which we observed in the stem. And the explanation for this is that there is a significant storage in the stems that is feeding the transpiration early in the morning, but it has its own seasonal cycle. So it allows the plants actually to avoid the atmospheric stress of the midday high VPD. And then we did a manipulation experiment in the forest where we irrigated one plot during the summer. So we maintained the soil moisture constant throughout the year, trying to maintain it at the same level as in the wet season. And that was a tool, again, to try to partition VPD and soil moisture because in the summer, both plots were exposed to very high VPD, as I showed you in the previous slides, up to 6, 7, sometimes 8 kilopascal. But we eliminated the soil moisture stress. And you can see here the soil, the extractable soil moisture content. It goes up in the winter, it goes down very sharply in the summer, but in the irrigated plot, it was maintained more or less at the same level. At the same time, VPD is going up to these extreme values of 5, 6, and even higher kilopascal in the summer. And what Yakir observed when he looked at the gas exchange measurement is the transpiration in the summer actually went up way above the levels in the wet season. And this is the effect of VPD, as we will see in a minute, because soil moisture is not limiting, but VPD now in the summer is very high. Stomatal conductance remain generally high, even with some spikes throughout the summer when water was available. And assimilation also continued the same level of as in the wet season. You will notice the big effect was on the evapotranspiration, where the water that allow conductance to remain high and assimilation go maintain high, but here the VPD of the summer, the very high VPD, actually enhanced the evapotranspiration in this treatment. And indeed, what he observed when he looked at transpiration as a function of VPD, in the irrigated plot, there was almost a linear response of increasing evapotranspiration in VPD way beyond the values that are observed in temperate forest, where it is assumed to cause a shutdown on stomatal conductance. We didn't observe this in Yakir. You can see also the stomatal conductance increase in the irrigated, the summer irrigated plots, but remain constant across most of the range without preventing the increase in transpiration. So it's really a pretty direct effect of VPD. As I mentioned, Yakir moved on to Harvard to do a postdoc, and he did some experiment there under control conditions with 2 students there. And they did a similar kind of manipulation as we did in the field in Yakir under controlled condition at Harvard with modifying the soil moisture and the VPD. And essentially, they got with very different species of plant. This is grapes compared to the conifers, the drought-resistant pine forest in Israel. Essentially, they got the same results with transpiration correlating essentially linearly with VPD and increasing with the soil moisture going from red to blue. So this is a reminiscence of what we observed in the field in Yakir. So the conclusions that we drew from these studies, or Yakir drew at this stage, is that VPD function as a driving force of transpiration as long as water supply meets the demand. More complicated measure, to measure, but soil moisture and water potential are better predictor for transpiration. By the way, water potential was not measured in our experiment, but it was modeled, and it was, at least in the modeling, we could show that water potential is really the key to maintain stomatal conductance, and water potential remained reasonably high in the experiment that we did, at least in the model. Now we have some additional water potential measurement that actually confirm this. That is the case in pines and in grapevine, which are very different in terms of physiology and adaptation to drought. but, and therefore we think that it might be a general phenomenon. Under warming climate, plants will regulate their gas exchange according to water supply, a factor that should be better predicted in using the models. I think with this I will end, and these are all the collaborators that Yakir has, both our lab and Jose from the Hebrew University and Holly from Harvard, and all the exciting students that we have in the group. Thank you.
Until some threshold at which you reach the hydraulic capacity of the tree. And especially when you begin irrigating, you know that these trees started to grow and these trees in the heat area. I don't think they were ever at a steady state where you reach that threshold. So it's okay to show the results, but you didn't reach that threshold.
Yeah, actually, surprised that we couldn't reach the threshold. There were some indicators. You know, you can imagine that maybe here things started to get complicated, and we thought that this might be where the capacity of the system has been reached. It's not very clear from this data that this is the case, but you're right. At some point, we should reach the capacity of the. We were surprised that this went up so high because pine trees are considered to be very conservative in terms of conductance and both xylem conductance and stomatal conductance. So, but as you say, these are.
Actually, if I could, so when we compute irrigation requirements, so we compute according to there's no threshold.
Okay, so, yeah, but yeah, there is a problem with the Perelman-Montis problem that is no limit. I don't think this will go on forever. You know, there is a limit. It's simply that it's not very clear in Yakir's results that we reached it. So I didn't consider it as, you know, it's not clear. But it might be that this is what you're referring to. This is, yeah, we need global change and increase in VPD to continue to to reach this point. Yeah. Yeah. So you're more brave than we were in concluding this, but yeah, we will wait for a few more years and extend the line.